Thank you. So today I'm going to talk about one of the final steps in the histology pipeline. So this is to use electrophysiology features that you have recorded on the probe to help align the track in the atlas and get an accurate estimate of where your electrodes are basically in the brain, so where you've been recording from. So I'll first give a short introduction to the topic and the concepts, and then I'll go on to show a demo of the tool that we developed within the International Brain Lab for this purpose. So the first thing that you need to do is, I think Stephen touched on this, but is to basically get an initial estimate of where your channels are located in the brain. So for this, you need your histology image, and you need to register this to the Common Coordinate Framework or the reference atlas. And this involves like scaling and warping the brain to match this reference template. And once you have that, hopefully if you've labeled your probe correctly, you should have a nice fluorescent track that you can then scroll through the image stack and trace where your probe was in the histology image. And this allows you to get a 3D reconstruction of where your probe was in the brain. And then you can use the initial geometry that you know from your probe. So for example, for the Neuropixel 1.0, you would place the first electrode 200 micrometers from the tip of your trace track, and then distribute the remaining electrodes uniformly at intervals of like 20 micrometers.
Mayo, I suspect at this point, 5 out of 194 people are wondering, why is this not a straight line?
So I think this is due to manual tracing step. So as you scroll through the way this probe was traced was in Lasagna, so the software that Stephen mentioned earlier. And their idea is you manually click on the trace, scroll through the stack, click on the second trace, and then it basically creates a line of best fit, or kind of like a curved line of best fit to those trace points. So that's why it's not a completely smooth track. So once you have your initial electrode locations, because we are in this reference volume, each XYZ position is associated with a voxel that has a brain region associated with it. And so in this way, we can then get kind of an image like on the right, where we can see the regions through which our electrodes pass. So this kind of gives you a first estimate of the electrode locations, but as Stephen was mentioning earlier, I wouldn't trust these ones because there's a lot of uncertainties here. And so these arise from the fact that you have subjects with different brain sizes and different distributions. So when you warp and scale these to match the reference templates, different regions will be scaled differently. And so we need to account for this. And also there's inaccuracies when you're tracing. So at the moment, at least within the IBL, this is a manual step. And so if you don't accurately locate the tip of your probe, then you can find all of the regions are shifted slightly. And this is also made worse by the fact that the dye sometimes diffuses to local tissues. So where you think was the tip might not actually be the location of the tip. So to get more information, this is where we bring in the electrophysiology that we recorded on the probe. So this schematic just shows a simplistic view. So on the left, we show an initial estimate of where we have the brain regions where the electrode passes. And then on the right, we have the firing rate recorded on the probe. So we have this white area here. Can you see my mouse? You can, good, okay. So we have this white area here, which represents just a kind of a ventricle or an air gap in the brain. So obviously in this area, we wouldn't expect there to be any neural activity. And what we can do is apply reference lines on both the histology and also on our electrophysiology features. And we can use these to basically shift where we think should match and shift the location of our electrodes. So in this case, we've shifted the electrodes up in the brain so that this area where there is no firing rate matches the air gap. So this shows a very simplistic alignment where all we've had to do is apply an offset. In reality, we want to be able to scale the electrodes along the probe and also within certain regions. And so for this purpose, as part of the IBL, we've developed this tool, which can be found at this GitHub repo. This basically allows you to show a number of different electrophysiology features alongside histology and use this concept of reference lines to basically move your electrodes along the trace track. So I'll give a demo of this, but before that, I thought I'd go through a number of a few key alignment tips, so useful features that you can use in your alignments. So the first is white matter and ventricles. So these are denoted by gray and white areas in the Allen annotation. And basically in these areas, we don't expect there to be any activity or much activity at all. So this figure shows the spike raster across the duration of recording along the depth of the probe. And as you can see at the top and the bottom of the probe, we have these blank regions, and these correspond to these white matter areas. And this can also be seen if you look at the firing rate across the session. Another useful region is the dentate gyrus. So this has a very high power in the LFP band, so in the low frequency band. And here we show basically the power spectrum overlaid on the geometry of a probe. And you can see in the dentate gyrus region, we have a very high power at that region. And the dentate gyrus also has these molecular layers that sandwich the structure. And these are also useful as the molecular layers tend not to have any activity. So you can see that we have these quiet bands that surround the main dentate gyrus, and the molecular layers are often silent. And the third useful kind of thing to look out for is pyramidal cell layers. So these typically have neurons with very high firing rate and amplitude. And so if you look at, for example, the firing rate view, you can see we have a peak here. And in this case, it corresponds to layer 5 in the cortex. And similarly, in this example, we can see that we have high firing rate in the CA1 and CA3 layers of the hippocampus. So I'll now stop sharing this and go over to the GUI to give a quick demo of how you can use it. So this shows how the GUI will appear once you've loaded in the data. So initially, when you launch the GUI, you'll see a blank window that appears. And what you need to do is navigate to this button that has 3 dots. You click on it, and then you navigate to the folder on your local file system that contains the data. So in this case, I'm just using the sample data that I think you may have already downloaded if you installed the tool. So you select the folder, and then you hit the Get Data button. And it will then, it takes a bit of time, so that's why I've loaded it in advance, but it will then load in these plots. So the GUI is split into 2 sections. So on the left-hand side here, we have all of the electrophysiology plots. And in the center here, we have the histology plots, which show where your electrode is in the brain. So in the electrophysiology plots, we have 3 panels. So we have these image plots that show, so in this case, we've shown the firing rate across the duration of the recording along the depth of the probe. And here you can change the view of the plot. So we can, for example, look at the cross-correlation across depth, the activity, the power spectrum of the LFP band between 0 and 300 Hertz. And we also have a number of these cluster plots, which show the location of the clusters along the depth of the probe. And here they're showing the amplitude in the X, and the colors denoting the firing rate. And in these plots, you can actually click on the individual clusters to get to view their waveform and also the autocorrelatogram. That's a useful thing to look at. In the central panel, we have a number of line plots. So these just show average activity across a session. So here we've just got the firing rate, and you can also see the amplitude. And then on the right panel, we have these, we overlay features on the geometry of the probe. So in this case, we're showing the AP band of the RMS. RMS of the AP band. And there's a number of different options. So you can look at like different, the power spectrum at different frequency bands. And here in the central panel, so we have 2 histology images. So these show the initial estimate of the regions that the probe passes through. So the left one is going to change as you align, and the right one is just there as a reference. So you can see how much your alignment has changed compared to the original. And over here, we then have this an example slice for the histology, and overlaid are the electrodes. If you zoom in, you can see the individual electrodes, and you can see where the probe was traced. So everything in the alignment tool is based on reference lines that I referred to earlier. So these can be added on the tool by double clicking in either the electrophysiology plots or the histology plot. So you then get a reference line, and these move independently in this panel and in this panel. So I'll just go through and align this how I would do it and describe the process that I'm going through. So the first thing that I would do is to take this region where we see it end in activity and denote that to the top of the thalamus. Once you're happy with where your 2 lines are, you can hit the fit button, and you'll see that your channel locations shift. And I should say that these dotted lines at the top here, these denote the top and the bottom of your recording electrodes. So if you focus on this histology image here, we can go back to before and after. we've applied that alignment, and you can see that the channel locations have shifted up in the brain. So the next thing I'll do is probably use what I described earlier about the molecular layers of the dentate gyrus, and use the say that these silent regions probably correspond to those areas. So I'll keep adding some reference lines and align them. And as you do more fits, you'll see this color bar appear in the center. And this basically indicates how much scaling you're applying to each region. So you can hover over this, and in the title here, it will tell you the scale factor. So here we've scaled these regions by 15%. And you can hover over each region. So then the final alignment that I would do will probably be to align this high amplitude peak with the layer 5 of cortex. So you get something like that. So in histology, you can also change the view. So in this case, I'm showing the red channel of histology, but you can also show the green channel, the reference atlas, and also the annotation atlas. And you can zoom in to see where your reference lines are and see if they roughly look to be in the correct location based on these boundaries. And once you're happy with your fit, you can then hit this upload button, upload your alignment, you confirm. And then in your local file system where you had the data, you'll get a new file called channel locations. And you can load this, and basically for every single channel, you'll have the XY position in space and the brain region to which it's been assigned. So there's a number of other features. So there's a lot of shortcuts and other views that you can see. But one useful thing is you can also, so at the moment, this is showing all units detected by killer sort. You can also filter just by the ones that have been classified as good, or just MUA if you want to look at the bad ones. So you can also filter by those as well. But so on the wiki, there's a detailed usage instruction, which has a link to all of the, or explanations of all of the shortcuts and different control shortcuts and other things that are available. So you can look through that to see all of the things that are available with the tool. So I'll now go back to the presentation and talk a little bit about how you can prepare your data to be able to be used with this tool. Oh, hold on. Okay. So basically, the tool reads in from a local file system, and all of the files that it needs need to be in one folder. And the files that it expects follow the convention that we use within the International Brain Lab. So in order to use it, you need to basically transform the output from killer sort to have the same namings that we expect. So what you would need to provide is like just the spike sorting output. So this could be from killer sort or killer sort. And also you need to have access to the raw ephys data files. And at the moment, the converter that we provide is only compatible with SpikeGLX, but it should, it's possible to write it for other acquisition modes as well. So we provide a function that's called extract data. And basically, if you provide your killer sort, the output to your killer sort data, your path to your raw ephys data, and then the output path, which is where you want to save the data, you can run this function, and then it will provide, basically, it will change the naming of the spike sorting to match what's expected. And from the raw ephys data, it will also extract the RMS of the AP, the LFP, RMS, and also the power spectrum, which are useful displays on the electrophysiology features. And the other thing that you need to provide is so the coordinates of the track, basically the probe track. And I think a lot of the software provide this ways. So I know like Brain, Reg, and Lasagna, they output in Allen's CCF space. And actually in the tool we decided that somehow the IBL has decided to use a Bregma-centric space. So we provide some just helper functions that allow you to convert from CCF coordinates to our Bregma-centric coordinates, so that it can then correctly identify and show the regions in the brain. And the other option, so the ones that stars are kind of mandatory data sets that you need to provide. And then you can also optionally provide the histology image stacks, which allows you to view the first two histology images that I show, so the red and the green channel. It can also work without that, but you just see the reference CCF and annotation atlases instead. And the only specification for those is that they need to be in the NRD format and registered to the Allen reference atlas. Yeah, so here I just summarized a bunch of useful links to the code base, the main wiki that we have, and then the install instructions, which I think a lot of people have gone through, hopefully, to install the homework, basic usage instructions. And then there's also a more detailed section about how to prepare, which builds from the slide previously, about how to prepare your data for your data to be able to be read in using the tool. Yeah, and with that, I just like to thank people of the International Brain Lab, particularly Olivier, Winter, Gary, Nick, and Carol for their support and for helping develop this tool, and also Dave Yu and Han Hu, who have provided really good feedback and also helped me extend it to work without kind of outside of the IBL structure, infrastructure. And finally, our funders who have allowed me to do this well. Yeah.
Great, thank you so much, Mayo. You have to imagine 195 people clapping now. So there's a few questions and you were nicely on time. So I think there is time to answer them. And then when you're done answering the questions, do you mind copying the links that you had in your talk and putting them in the chat? Because otherwise people won't be able to click them. Okay, so question number 1 from Jing Yan. Does this EFIS GUI take the data directly from the kilosort output or from the phi output files?
So this is, from the kilosort output files.
Yeah.
Yeah, but they also then need to be renamed. But I think actually one thing that we did notice was that if you're going to do reassigning and merging, it's best to do that within the kilosort output files and do that first, and then convert. No, sorry, the other way around. I'll come back. I'll find there's a problem with this. I need to find out, but one needed to be done first in order for it to still be compatible. So I'll confirm on the Slack channel about that.
Okay.
Yeah.
And I suspect that you're not using phi output because we're not using phi. We're going straight from kilosort to data in the IBL right now because we have so much data that we can't possibly.
Yeah, exactly. So it is just, this is directly from kilosort. But I think someone ran it through phi, had merged the cluster, then tried to do the converter to make it compatible with the GUI, and they ran into problems. So I'll just check on that.
Okay, so Nick, do you mind following through with the questions? I'm going to make sure that Anne Churchland can log in.
Sure. I mean, Mayo, can you see the Q&A? Do you want to just...
Oh, yeah, sure.
There's one question about the specific input file types for the GUI. I think that maybe you've addressed that, or it would be best for them to read the documentation about that.
In the wiki, there's also a section that describes overview of data set. That describes all of the inputs expected. So it's kind of a description of what they are, the format they need to be, and the unit. So you can also refer to this to make sure you have the correct data sets.